In this video, we're going to look at how our body can repair some DNA damage. So typically in any given mammalian cell in your body, you have 100,000 molecular DNA damage that happens in a given day. This can be from your environment or from your natural metabolism, but this has to be repaired, otherwise your cell won't be able to replicate or create new proteins or do much of anything with this DNA. And generally, for every type of DNA damage out there, we have a specific enzyme that will take care of that for us. For example, if we get that TT dimer from UV radiation that I talked about in the last video, we can repair that with a photolyase. Photolyases are a monomer, and you have two prosthetic groups that go with them. You have a cofactor that will absorb light, and you have FADH. And the idea is this protein absorbs light by that light absorbing cofactor, gives that energy to FADH. Then FADH can cause the thymine dimer to split, and that's how we can repair that thymine dimer. And the way it works is that you have a very positive region on this enzyme that binds DNA, and you'll flip out the two thymines. That's what's shown here. We kind of just take the two thymines and we flip it out of the helix, so it binds in the cavity of our enzyme. And this is called base flipping. And then once we have it flipped out, we can break the bond between the two thymines. Now, when we flip something out, we just don't want to leave an empty space in the DNA, so the protein itself has a region that will go and fill in where the flipped out bases were. Another example are alkylotransferases. These will fix the base methylation problems that we talked about in the previous video as well. So what do we do if we come across some type of damage that we don't have an enzyme for and we can't fix it? What we can do is called base excision repair, and these are carried out by glycosylases. What happens is that a glycosylase will come and it will cut the glycosidic bond of the altered nucleotide. So you're just left with a sugar at that part. This is called an apurinic site or AP site or abasic site, where you just have a sugar but no base. Then we have AP endonucleases that will come and will cut the sugar off to just leave nothing. So that'll be completely gone. And you can also, and when that's gone, what will happen is that we need to replace that base. So we're just going to cut away some of the bases around it by an exonuclease. Then polymerase can come in and fill in that gap, and ligase can seal the nick. So it's kind of like mini replication event, because we will have our template to know what should go on the other side. So let's look at a specific glycosylase here, and these are urinal DNA glycosylases, or UDGs, and these will get rid of any uracils in your DNA. So this could happen from cytosine deamination or an accidental misincorporation of uracil instead of thymine. And what it does, it's a lot like the last enzyme we talked about, is that it's going to bind DNA and then flip a UG base pair out of the helix. So, for example, this would happen if your cytosine turned to U, you would have a UG base pair, and we flip that out. You then cut the glycosidic bond that attached your uracil to your deoxyribose, and the uracil in the AP site will remain bound to this enzyme. So you just don't cut and go, you just kind of keep it bound. And the reason is that these AP sites are toxic. If topoisomerase 1 came along, it would get stuck on this abasic site and would never be able to leave, which basically means that cell is condemned to death, because your genome could never be fully replicated. You would have a protein just stuck on your DNA forever. So to prevent that from accidentally happening, these uracil DNA glycolysis will just hang on until it can come and be fixed. So where that base was, arginine 272 on our protein, we'll go and just stick in that site, and that's what's being shown right here. And the way that these glycolysis, there we go, work, is that they're looking for specific geometry. So if you have a UG base pair, your DNA is going to have a kink of roughly 45 degrees. This protein binds to that specific structure. That's how we can recognize a UG base pair is present in the DNA. And this protein only works with uracil. So even if there was another region that had the same geometry, it wouldn't cut unless there's a uracil there to prevent any accidental hydrolysis of DNA bases. Now, another type of repair we can do is called nucleotide excision repair, and you'll often see this written as NER. So let's say that you have some DNA damage from sunlight or tobacco, and you want to fix this. One way we can fix this is to excise this portion of the DNA. That's called nucleotide excision repair. It's carried out by three proteins. UVRA, UVRB, UVRC, they work with ATP, and they're called the UVRABC endonuclease. So they're going to do the cutting, and what they do is that they're going to cut 7 nucleotides and 3 or 4 nucleotides away from the damage. So 7 nucleotides on one side, 3 or 4 on the other side. And when you have this cut, a different UVRD, which is a helicase, will come and just remove this cut sequence. Then once you have this cut sequence, Part1 or DNA ligase can come and just refill this gap, because you basically have an open 3' now, and you are ready to go. You have got rid of that lesion. So there are some disease states that you can get if you don't have these proteins. So xeroderma pigmentosum XP is if you don't have or you have a defective NER. So you can't really fix UV lesions. So you're extremely sensitive to sunlight. And this will show up when you're an infant. You'll have really dry skin, freckling, and a much higher case of skin cancer. And there's also CS, which is another genetic disease where you don't have a proper working NER. Again, you're really sensitive to UV. You have stunted growth, some neurological dysfunction, and premature aging. So the NER is critically important to maintain healthy cells, as we can see from these diseases that happen when you don't have NER. With that, that's the end of this video. I will see you in the next one.